Again, a pleasant day to all and to you, sir. Now, this is my second video presentation, and this is all about dark reactions. So, dark reactions, also called the Calvin cycle, the dark reaction starts and ends with the same product, hence the word cycle. All the dark reaction takes place in the stroma of the chloroplast. The Calvin cycle starts with RuBP molecules or ribulose biphosphate molecules and carbon dioxide molecules. An enzyme called Rubisco combines them into an unstable intermediate. This is the reason plants take in carbon dioxide to start the Calvin cycle and begin the conversion of RuBP into glucose. RuBP is the starting molecule and ending molecule of the Calvin cycle. It will be remade at the end of the cycle so that the cycle can begin again. Since the intermediate of combined RuBP and CO2 is unstable, it quickly splits in half to form two molecules of 3-PGA, which are stable. The ATP and NADPH from the light reactions provide the energy to convert the two molecules of 3-PGA into their final form. The leftovers are used in the light reactions to remake ATP and NADPH. Two G3P are joined to make a glucose molecule. Not all G3P is made into glucose. The Calvin cycle occurs in every stroma, in every chloroplast, in every plant cell, every second of every day. That's a lot of reactions all happening simultaneously. Most of the 3GP made during the Calvin cycle are made into RuBP or the starting molecule. With energy from ATP molecules, the cycle called the Calvin cycle can begin again. Thus, the spent ATP from the reaction leaves ADP and a phosphate group. These are used in the light reactions to make more ATP. Let's have a summary about dark reactions. The Calvin cycle converts the carbon from the carbon dioxide into glucose into the stroma. This is called carbon fixation because carbon is fixed into another form. In hot and dry environments, plants lose a significant amount of water through the stroma, which causes them to become dehydrated. In order to stop this water loss, these plants close their stroma, which causes continuous photosynthesis in these plants. Oxygen is produced by photosynthesis, which consumes carbon dioxide. Usually, this oxygen diffuses out to the open stomata. However, when the stomata is closed, oxygen builds up and carbon dioxide decreases in the plant leaf. Reminder: the chloroplast has three membranes: the outer membrane, the inner membrane, and the thylakoid membrane. Now, let's discuss the implication of an increase in oxygen within the leaves. The compartments include the stroma and the thylakoid space. With an elevated oxygen concentration, Rubisco in the stroma will attach oxygen rather than carbon dioxide onto the ribulose biphosphate. After the attachment of oxygen and the ribulose biphosphate, two molecules result: 3-phosphoglycerate, a 3-carbon molecule, and phosphoglycolate, a 2-carbon molecule. Phosphoglycolate cannot continue in the Calvin cycle. Thus, it is broken down and these carbon units are released as carbon dioxide and are wasted. Due to insufficient carbon in the Calvin cycle, G3P cannot be produced. However, the cycle does continue and the ribulose biphosphate is regenerated. This process is known as photorespiration. In summary, photorespiration wastes carbon, requires energy input, and does not result in the formation of G3P, a precursor of glucose. That's all, and thank you.